All right. Well, thank you for the invitation to present at this Teach Me in 10 talk, where I'm going to walk through some opportunities for accelerating cell and gene therapy, or CGT, by bridging the gap between in vitro and in silico research. So let's talk about some challenges in the cell and gene therapy space. There are several things that are top of mind at the moment industry-wide, and this includes the research community as well. There is the risk of safety, immunogenicity, and adverse events. This covers everything from the early discovery stage through the clinical trials. So risk of safety is going to be really primarily due to the delivery vehicle, which is very often viral these days. Immunogenicity is a challenge for both the virus carrying the gene therapy or cell therapy cargo and also the cargo itself. So nucleic acids can have certain sequences and motifs that may raise immunogenic responses, and so that's a challenge. And then adverse events, of course, are also largely due to immune system responses. Another major challenge is the high cost of goods requiring downstream enrichment, which is a very business way to say that it's extremely expensive to develop the gene therapy and cell therapy at the design stage, but it's even more expensive when you have to think about manufacturing and also enriching your manufactured product for something that is going to be safe and less immunotoxic, et cetera. A third issue is target tissue efficacy. So target tissue efficacy is describing the challenge where you've got to get the therapeutic to the right tissue. Most of the time, a cell and gene therapy is affecting a very particular part of the body where there is a protein or gene that has gone awry. And so getting the new treatment to the right place in the body is a pretty important challenge. And then lastly, and I alluded to this previously, is the design for manufacturing. So how do we make enough of this therapeutic that we get enough doses to cover a large enough patient population to be meaningful for the cost of manufacturing? So I want to spend a lot of time on the slides. This is a busy slide, but this is where I want to talk about where the opportunities for acceleration are. And by that, I mean reducing the churn on the bench to arrive at solutions to the challenges I just talked about. And this is a place where there are increasing numbers of in silico or computational tools to aid in that process. So let's just think about the process of developing a cell or gene therapy drug. So we start at the discovery stage, which very often is happening in an academic lab still, and we're identifying the genetic basis of disease. And so the opportunity for in silico tools here is really jumping up for laboratory automation, which from a computational perspective really means rapid collection of data and transformation into a digital repository where it can be rapidly affected. And then also digital model organisms. You'll hear a lot of digital twins these days, and that can mean anything from a lab environment itself and the equipment in it to the patient population itself. Digital model organisms describes the concept where you have a mouse model, a non-human primate model, sort of digital twin, where you can iteratively figure out what things are, like what the phenotype is likely to be if you modulate a particular gene and things like that. A lot of this wouldn't even be possible without a really large amount of whole genome data, and that is increasing all the time. And lastly, genotype-phenotype relationships, which are the major important thing that we have to figure out when we're trying to discover what the gene target is going to be. So this is a place of discovery where there are a lot of opportunities for cell and gene therapy. You see a lot of that happening right now in the small molecule stage, where we're using things like AlphaFold and other computational tools on the protein side of the biological question. And so for cell and gene therapy, things are really just starting to ramp up at the sort of gene and nucleotide side. Clinical development, this is where you're going to be identifying your lead drug candidate, so you know what the gene is. Now we have to make the therapeutic appropriate. So again, safety and immunotoxicity, et cetera. We're going to design the capsid, or we're going to choose a capsid that has a tropism for the tissue that we're trying to reach. Vector design, that's going to include what promoter you're going to use, if you want an enhancer, whatever you need to tune the expression level of the gene that you're delivering. And then, of course, optimization for safety and efficacy, where I live, Form Bio, that's where we're starting to really play. So the next up is candidate selection, process, and analytical development, which is a long way to say we figured out what our lead is, and now we have to optimize it for manufacturability. And we need to get enough doses out of it that it can be useful. We need to make sure that we're producing and purifying it enough that we're, again, taking care of safety and immunotoxicity, and then fill and finish, which just means how are we formulating this to actually give it to a patient? Is it a patch? Is it an IV? What is it? And then the last step, of course, is clinical development. And this is where we have to think about optimizing the process for GMP. And we have to think about things like patient and clinical trial matching, and then, of course, data analysis and outcomes reporting. And this is another place. So discovery and clinical development are places where there are a lot of tools that are up and coming. And preclinical development, especially the manufacturing process, is more recently understood as a major opportunity. Okay. So as I mentioned, where we live at Form Bio, we are thinking about manufacturability. So there are several places where you have to think about manufacturability because you're going to manufacture at different scales these cell and gene therapeutics across the timeline. So at the discovery, safety, efficacy stage, the safety toxicity stage here at the in vitro side into in vivo, you've got a lower scale. And then when you get into clinical trials, suddenly you've got this massive shift in material that you're going to have to meet. And so each of these stages are places where there is a lot of bench iteration. Just the whole process for one iteration can be like 2 to 4 weeks, right? Not including the synthesis. So that's very slow. And if you have to do it a lot, you can imagine how it would be really helpful to be able to speed this up. And so that is the design stage. The manufacturing stage, you have the same thing. You have so many variables that are very often right now manually tested in the lab to try and arrive at a good enough process that you can make enough doses to be useful. So what we're doing at Form Bio is we are leveraging large language models by thinking about cell and gene therapies as a language. So what I'm showing on this slide is kind of a graphical overview of the whole process, where we're starting with a sequence of nucleotides, which makes up all of the genes. And that's whether you're using a cell delivery mechanism or a viral gene therapy mechanism. At the end of the day, we're starting with nucleotides. And nucleotides are letters that make up a language. And in a construct for a therapeutic, we have annotations, which are a language on top of that. So you can see on the left, we've got a promoter section, we've got a CDS, which is the coding region for the gene of interest that we're delivering in this example. And so these things make up a construct that include ITRs for viral packaging. We've got a promoter and a gene of interest and a poly-A tail just to get the expression cassette built. And then we have to match that up with an appropriate rep and cap gene so that we can package those into the viral capsid for delivery to the eventual patient. So we've got the patient genome, which is going to be the human genome most of the time, unless it's a cell therapy, in which case we've got personalized information. But that's also reducible to all of these different gene sequences and chromosomes and things like that, right? So at the end of the day, it's a language. And so we are taking advantage of that by using large language models to predict motifs, types of secondary and tertiary structures that are going to form, to identify places where we've got opportunities for correcting possible issues, truncation events, things like that for manufacturing before we have to do a whole lot of bench iteration. And so the way that we are doing this is we've basically built our entire AI ML deep learning process and delivery engine, and we're using a transformer neural network as our major LLM component. We have pre-trained models for coding and inference that we are updating all the time. And then, of course, machine learning models for identifying the different tasks that we are using with our entire model framework. And then on the right, I'm just giving you an idea of the scale of the data sets that we are collecting. These are next generation sequencing data sets on actually viable AAV gene therapy candidates. And so the more data we have, the more we are able to predict, the more variety we see, and the better we're getting at being able to predict these events that are going to be problematic for manufacturing. And this last bar graph at the bottom is just showing we're doing a lot of bench validation right now, where we take our constructs that we that we designed with our model. And then we go back to the lab and we see, well, how well did they do? And so you can see here in green, that is our NGS, next generation sequencing validation data. And in the blue bars are what our model predicted for those sequences. So this is a really important part of speeding things up. Is it's really, you've got to believe that the in silico tools that you're using are going to be reasonably replacing the churn in the lab. And that is the way that we're going to get faster. We're going to get faster at every step of this process. And I will thank everyone for your attention.